Dear students, now we are going to discuss a very famous and very important model of DNA that showed that DNA exists as double helical structure. And this very famous model was presented by James Watson and Francis Crick in 1953, before anyone could ever explain about the structure of DNA. They put forward their model. And according to their model, DNA existed as double helix. Double helix means there are two helical strands that run across each other, pointed towards each other, and in an anti-parallel fashion. Means they are parallel, yet their direction is opposite to each other. One strand is 3 to 5 and other one is 5 to 3. So the strands, they are anti-parallel to each other. The bases that are present, they pair in such a way that always a purine, it binds with a pyrimidine. And cytosine, it always binds with guanine, and adenine and thymine, they always bind together. There exists a double hydrogen bond between adenine and thymine, and a triple bond between cytosine and guanine, and this bonding is highly specific. So since A cannot form bond with C, so this bonding is not possible, and vice versa. This smaller groove, you can say, this is known as minor groove or the small turn, and this is the major groove or the larger groove. This is known as the major groove. The distance between two turns is 3.4 nanometer. And the distance between the base pairs, this distance between two base pairs is 0.34 nanometer. So in the model presented by Watson and Crick, base pairs always consist of purines, which are large, pointing towards pyrimidines that are small, keeping the diameter of the molecule constant. Because of the bonding between these nitrogenous bases, the DNA becomes twisted and coiled. And also the DNA strands, they are running in a complementary fashion to one another. In the double helix, the adenine, it always forms 2 hydrogen bonds with thymine, while guanine, it forms 3 hydrogen bonds with cytosine. Adenine will not be able to form a proper bond with cytosine. Therefore, this bonding is not possible. And consequently, adenine and thymine will always occur in same proportions, and cytosine and guanine, they will be present in equal proportions because of this base pairing. So this was all about the structure and composition of DNA. I hope you have understood very well. Today we have discussed about the chemical nature of DNA. We found out that DNA consists of nitrogenous bases, phosphate groups and a 5-carbon pentose sugar. And DNA is composed of small subunits that are known as nucleotides. The nucleotides, they join together to form macromolecule of DNA. Then we have studied that DNA model was presented by James Watson and Francis Crick. So this model showed that DNA exists as double helix coiled around each other, two chains that are anti-parallel to one another. They are coiled or twisted around each other to give a double helical shape to the molecule of DNA, and the bases, they are always bonded in a specific manner. A always binds with T and C always binds with G. The distance between two turns is 3.4 nanometer, and the distance between the base pair is 0.34 nanometer. There is a minor groove or the smaller turn and the major groove or the bigger turn. So this is all about DNA. The topic of DNA is not closed yet. In the next lecture, we will be discussing the DNA replication, which is a very important phenomenon because it is required in order to copy the genetic material to transfer it to the next generation. And if DNA replication could not occur, there will be no transferring of genetic material from one generation to the next generation. So our next topic of discussion will be DNA replication and its experimental verifications, and we will discuss in detail the replication process. Till then, stay tuned, keep watching. Thank you so much for your time. Allah Hafiz.